Yes, sir. You can start now.
Okay. So we have good evening, everyone. Welcome to friends, colleagues, and students who have joined the platform. We are in the second lecture, which is organized by GIL. Is anybody saying anything? No. Okay. So the GIL is actually Genome India International is a platform which is trying to do good for all those scientists who are working on genome, which might include genetics, breeding, genomics. So all those people are being brought together by Genome India International. This is an organization which started in 2005. And Dr. Chitanjan Kohl is the president of this organization. And then there are people who are trying to contribute to the platform and bring forth more and more information to everyone. The platform's whole purpose is simple, so that all the people come together who are working on genomic science, interact with each other, if possible, mentor the next generation. So we encourage all those who are working on genome science to join this platform, whether you are a student, a scientist, a breeder, so that we all can work together in a synergy and try to bring forward Indian genomic science to the world level. We have very good people who have been contributing to this platform. And one of the initiative taken is actually to provide lectures. And the lectures, people who are selected for the lecture are at the forefront of their field. The first lecture we organized was by Professor Christian Heller Foyer, which is from University of Birmingham. And now in the same series, we are doing the second lecture, which is by Professor Manish Kumar Pandey. He is a principal scientist in ICRISAT, Hyderabad. I will be giving you more details about him. But before that, a simple request about GIL. Please join our program. Our program is very simple and straightforward. We are not looking after too much of money. Even the membership is kept very low, because we want more and more people who are working on genomic science to join us. And the idea is only that we all grow together and we interact with each other. We have a website. If you search for Genome India International on the Google platform, you will find the website easily. If you want any information, please contact us. If you have any program which you want to integrate into Genome India International, we'll be happy to do that. So we look forward to hear from you. Now, moving forward, I will like to talk a little bit about our speaker of the day. I will not take too much of time because I know you all are eager to hear him. We have Dr. Manish Kumar Pandey with us, who did his PhD from Osmania University while he was working with the Institute of Rice Research. Currently, he works on peanuts and the, sorry. So, okay, he works on groundnut and pigeon pea at present, and he's looking into genomics, breeding, and bioinformatics of the two crops. He did his postdoc from University of Georgia, and he's also adjunct faculty at University of Southern Queensland, the Shanghai Academy of Agriculture Science, China, and he has very good publication to his credit. He's been under 2% highly cited researchers from last 3 years continuously. His publications are in top journals. I can name a few: Nature Genetics, PNAs, and Molecular Genetics, Molecular Plants. So we are hearing from a person who is at the top of this field. He is a member of NAS, NASI, TAS. So I think if I continue with his CV, it will take more time than his lecture. So what I will suggest that we go to him and hear him out directly. I have been looking forward to hear it because peanut is a very interesting crop, and his talk today is about peanut, which is a tetraploid crop with diploid progenitors. So with no delay, Professor Manish Pandey, please go ahead with the lecture.
Thank you, Dr. Goyal, and thanks for this nice introduction. And nice to see like a lot of people here, and I can see many people are friends here. I can see Ved Prakash Rai also seeing, and then my own colleague like Prash, Dr. Panratna Parke I could see, and then Dr. Naveen Pupala also I have seen somewhere in the list. So thanks for this opportunity, and let me load my presentation to move forward. Please confirm once it is like properly comes. Yeah, can someone confirm like whether you can
Yes.
see the slides?
Yes.
Okay.
Slide available.
Okay, thank you. So again, like wherever you are, good morning, good afternoon in different regions. So as you already let Dr. Goel introduce me. So I work in groundnut and pigeon pea. In groundnut, I have been working since last 10 years. So you can say a decade. Pigeon pea, I started this year from this year onward. So just a beginning for me in pigeon pea. Groundnut, like we can say, most of you know that this is a tetraploid crop that we grow in farmers' field, and both it has two subgenomes, A and B, and both are really very similar. So many of the times it creates very much problem whether you are developing genome assemblies or you are doing the genomics analysis and then doing some practical applications for the breeding. Okay. So how we proceeded in last, so you can say I'm sharing the journey. I'm not giving any expertise or say any say lecture for the profit, but what I'm saying here for not for the benefit of knowledge, but you can extract the things as you like. What I feel and then happy about it that you do science, make something use of that science information and develop some technology that is useful for the end users. In this case, it's a variety. And then how we have used the genomics information to modernize the groundnut breeding program here at ICRISAT with international collaboration. So that is the story here today.
So
Most of
sorry, I think your slides are not changing. Have you changed it yet?
Yeah, I changed to another one. So
yes, sir, kindly make the presentation in presentation mode.
In presentation mode.
So it is not in presentation mode?
No, it is not.
One minute, please.
No, I think it is not still in presentation mode.
Yeah,
Namaskar, Manish. Usually, this happens with Google Chrome. Maybe you can see two windows. You could probably, click the other one, or maybe rejoin once. That would probably works. Sometimes.
Okay, so that maybe let me see.
Sir, go to slideshow option.
Slides option.
Slideshow.
On the top.
On the top.
It is the slideshow option. So you see animation slideshow.
Slideshow.
On the top, in the menu. In the menu.
No, so I have here.
Yeah, here.
I have mute options.
Sir.
Then stop video, participant chat, new share, pause, share, annotate, remote control, apps. I could see.
You have to go above that. It's written design, transition, animation, slideshow.
At the top of the presentation, sir.
The colored portion on the top.
Yeah. Home, insert, draw, design, like that. Yeah.
Okay, 1 minute. One minute, please. Give me 1 minute, just 1 minute.
Sure.
Can you see this slide? Not in presentation mode, but can you see the slide?
Yeah, we can see the slide, but it's not in presentation mode. So it is stuck at the presenter mode. They also have a presenter mode, I guess.
For not being able to share this. And then what we are going to share here, like journey for last 1 decade. And many of you may be aware about the CRISAT, that where we work in different crops, 6 crops majorly: finger millet, palm sorghum, and then among legumes, chickpea, pigeon pea, and groundnut. And I specifically work on groundnut since last 1 decade, and then pigeon pea from this year. We have good field here for phenotyping and other experiments, and also a good lab, genomics lab, that called Center of Excellence in Genomics and Systems Biology, where we have the sequencing and genotyping technologies. And if we see like production and productivity for groundnut across the globe, so total we have around some 54 million tons of production and around some 32 million hectares of area. But the productivity, if you see, that is very much good in USA and Argentina, and then you can see in China. However, if you see the area, actually India is having the largest area. So you can say that we have still a lot of opportunities to increase the productivity and then increase the production in coming years. So that's an opportunity. Now, as you know that not only in India, basically in Africa and many of the Asian countries, how groundnut is being consumed. So mostly like, okay, let's give example for India. If 1 decade before or 15 years before, 70–80% of groundnut was crushed for oil extraction, but and 20% only was going for some snacks and all those preparations. But now the situation is getting reversed. And now, as of now, we get around some say 50–60% of the produce for crushed, and 50% remaining going for confectionery. So you can see change. We are getting day by day, whether it's a peanut butter, whether it's like say it's different snacks with peanut. Sometimes you see chickpeas we mixed with, I mean to say, different kind of preparations are coming up. And you know, because of the nutritions, you see that it has been used actually by the different nations, especially in Africa, to provide them as like say ready-made food, nutritional food, to cope up with malnutrition problems. So even in India also, like most or many of the those primary schools where a meal is provided, this groundnut is used an important ingredient there. Now, if you see the climate change scenario here, and the red ones you see, actually it many of the times matches where the groundnut is being produced. Now, that means groundnut is going to face problem for the climate change for sure. And now we have to see that how we can breed this crop, which can withstand not only just for drought, but also for the heat tolerance in coming years. So basically 2 of these important traits. Okay, now let's just like invest like 1 minute of time in this slide. So this actually provides our strategy, how we are deploying genomics in the groundnut breeding program. And to do with that, we have genetic resources. That means we have germplasm like groundnut. Here at EquiSite, we have some 15,000 diverse germplasm here. And then what we do, we have phenotyped them and also genotyped, and then made a core set of diverse panel. That means this reference set is representing the core diversity for this global germplasm. So most of the times breeders and then genomics people, they use this one for phenotyping and doing different analysis and selecting genotypes for different traits to use them in their breeding program. For mapping, we know that we also do, because in plants you have freedom to make family-based populations. So we make genetic populations, you can say recombinant independent lines, or you can say biparental populations, and also multiparental populations like MAGIC and NAM, nested association mapping population. So that also we have made in this groundnut crop. Then again, you have like light cultivars, mutant populations, and of course, all these germplasm as much you can do phenotyping. So this is how like the genetic resources we have to use with very justification, or you can say precisely, in our breeding and genomics application. Now, to use the phenotyping data generated on this diverse germplasm, we need some basic genomic resources. And the first comes is we need to have one good reference genome, because that is like say basis for all our genomics analysis. Then of course it's good to have core and fine genomes. Then you have to do resequencing germplasm, gene expression atlas. It's good. You can estimate that what kind of genes are expressed, which tissues are useful. Then of course you get candidate genes, whether you do transcriptome, you do genomics, you do anything, you get some candidate genes for particular traits. You develop some of the markers, you validate for the trait. And then of course there are opportunities to develop different kinds of genotyping assays, low cost, because that is very important. If a breeder feels that phenotyping is more like say cost effective than genotyping, then it's very difficult to convince them to use the genomic tools. So that is how we have come up, how I will explain later. Now, what kind of genomics application? So most of the times when you interact with breeders or other like community research community people, you most of the times you come across only trait used for marker tested by crossbreeding. Actually, genomics application is not all about that. We see the application of genomics in duplication of gene bank, identifying those duplications, doing genetic diversity. Of course, you can do GWAS and genetic mapping and other genomic approaches for candidate gene discovery and marker development. You can use them in marker shed by crossbreeding. Also, genomic selection is one of the application has come up. When you are doing now, because most of times, like say breeders, they get away with saying that, okay, we don't want to use the diverse germplasm because it is not so elite. So again, you can do background selection with these assays, and then you can be sure that you are getting the background selection genome recovery faster with the use of markers to avoid genetic linkage, right? Of course, in breeding, many of the breeding programs like at ICRISAT. So we have started to genotype all the F1s to get make sure that all the F1s are true, and then only those will be selved for or backcrossed in further generation. And we have seen that maybe 20% we are getting efficiency. So basically, that is a good percentage. Now, again, there is one more application coming up very fast, that is getting seed purity check-in. Okay, seed genetic purity, where you can do in the parental lines, you can do for a variety in the seed system. So other applications coming up, because when companies will come, they want to purchase a particular variety and you have to fingerprint them. So there are opportunities at each stage, like from breeder to the consumer, how you can trace these varieties, all those things, fingerprinting is needed. So that is like another application coming up. Okay, now at this stage, what we are following that, okay, we go for genomes, fine genomes. I will be telling the journey about it. Then how we characterize germplasm, how we phenotype them, how we genotyped, and then went for gene discovery or haplotypes. And then after getting these, how we are developing this cost genotyping, like cost-effective genotyping assays, and then how we are using. So we'll give you some of the examples here. And ultimately, what we are doing actually modernizing the current breeding program. To give you a glimpse of like how this Arachis species at such like Arachis genus is diverse, you can see here. This is genus where you have Arachis, but you have different sections here, 9 sections sitting here. You can see how much genome diversity you see here. Different genomes. Within those genomes, species, you see number of number of chromosomes, and you can see like genome diversity here, haploidy level differences also. Now, where the cultivated groundnut is sitting, actually, this is under genus Arachis. Cultivated groundnut sitting with AB genome here under the section Arachis, and species Arachis hypogea. Now this is Arachis hypogea, which is meant like say considered that it is because of random cross between the A subgenome like A subgenome and B subgenome that have come together say some 10,000 years back or so, and then you have a Arachis hypogea. It is also been considered that in actually A monticola is also a tetraploid, and then maybe before coming to A hypogea, A monticola came and then it gave to the cultivated ones. So now we'll see what is happening, whether from these two diploids A and B, how this A monticola came, and then currently we have the cultivated ones. Now please also try to understand here currently what kind of ecotypes, you can say agronomic types or botanical types are grown currently. To understand it very simple, what we can say in Asia and Africa, mostly Spanish bunch type of genotypes are grown. So that is what we are seeing here. And then Virginia bunch and runner are the ones that are being grown mostly in US, Argentina, and Australia like countries. And that is why if you relate actually, what are actually differences between these? So runners are like they creep. So basically single plant occupies a lot of space, and then naturally their maturity goes higher, say around say 145, 150 days or so. And then yield is really very high. In Asia, especially in India, we grow Spanish bunch. That is a little bit like say V-shaped kind of plant. So not all the actually pigs that goes inside the ground and make the seed. So that's really, that is why you see, and it is short duration. So it matches to our like say cultivation practices here and all those seasons. So we are very much okay with the duration it has, and the yield scenario is also now increased. We can get in a like say given field approximately 3.5 or 3 tons per hectare. So that is how we have reached. So again, now the question is whether one genome is sufficient. So it's like the question before us, whether it's sufficient for us or we need more genomes. So definitely, actually, it is not necessary that only one will represent. So we have to see that can we genotype all the germplasm, make different groups, and from each subgroup we can have reference genome representing that particular subgroup, because each of us are not same. So we will miss out some of the genes you have, another person has, so it will be missed out. How we have captured will come up with information in next slides. Okay, to begin with, we were struggling to have first the diploid reference genome assembly. Initially, we deliberated at the like international level and then thought that, okay, let's go for tetraploid. But based on the discussion, we decided, no, let's first go to progenitor species, because that will be less complex. And the technology, you can understand, that was around 2012 and 13, like Hi-C and then PacBio and then like, you know, nanopore, all these technologies were not that much applicable. So, and that was the like say, like disadvantage to have reference genomes with those kind of technologies. So we started with those individual genomes, so that it becomes, when you go for tetraploid, it becomes very easy for comparison in aligning that. Okay, so we went ahead and then developed like genome assemblies for Arachis sapiensis, that is B subgenome donor, and also for the duralensis. So both we have developed. Also, there is one genome assembly came for Monticola also. That is thought to, that is considered to be wild tetraploid, like wild relative of tetraploid. So the cultivated ones now. So all these genomes came. Then if you see into that, this was in 2016, this deployed genome. Then we also got access to the other technologies like Hi-C, PacBio, and all those things. So when while developing the tetraploid genomes, so in parallel, 3 efforts were going on. And as I mentioned that we have 2 types of major, like 90% of the global area is covered by either Spanish bunch or Virginia bunch. So you can see that the first assembly that is coming, this is for say hypogea that we are talking about is Spanish bunch, and this one is for runner, and this is also for Spanish bunch. So basically what you can see, two for Spanish bunch and one for runner. So two types of assembly came, and technology you can see for each of these. High-C and PacBio were part of it. That has really helped actually in getting the perfect alignment. And once you have a good genome assembly, perfect alignment, your annotation also is good. And then that reflects all the analysis we do in our genomic analysis. So this is all the, like say, summary of the presentations. Right now, what we are doing now, we have even for ECRISAT, we wanted to have like ECRISAT specific reference genomes. So we genotyped the different groups, and we are trying to develop now this generating reference genome assemblies for these two genotypes, a new one, and also trying to completing near about resequencing some 2000 accessions, diverse accesses. So that is the longer plan to have analysis further in coming days. Now, what we are doing with the using the same tetraploid genome, we initially sequenced the 300 reference genotypes. If you remember, I mentioned that from 15,000, there is a core set, basically diverse panel of 300. So we use sequence that one, and out of those 264 are cultivated. So that resequencing data we used for different analysis. You can say structural variations, and also getting SNPs from tetraploid. Also, we developed pangenome for different subspecies and also overall also. And what you can see that to the reference genome, we added around some 82 MB region. That means that was missing from the reference genome. So you can assume that as many genotypes you resequence, this genomic region, the size will increase. So you are contributing actually diversity to the existing reference genome. So we are doing now this analysis for different traits. Because of time, I have not kept much information on this. After the reference genomes, pangenomes, so we developed gene expression atlas also for different tissues, some 22 tissues here, 20 diverse tissues, and then we have all this. So mostly what we do, when we do some genomic analysis, we identify some candidate genes. We use this gene expression atlas to see their tissue specificity. We can see that, okay, particular candidate genes we identified, how is the performance basically about the different tissues. So that information we get from this gene expression atlas on routine basis. Not only for subspecies hypogea that we are growing, also for A. glabrata, that is you see in a different wild, that is a different genome that is being developed with one of our Chinese collaborator, that is from SAS, Shandong Academy of Agricultural Sciences, China. So that is also become soon genome also, reference genome also will become available. Among the wilds, not in a Arachis species or section, this is like say glabrata is the one you might have seen. It is used as an ornamental plant. You can see flowering plants, it spreads, it never sets seeds actually. So that is the like issue with this one, but this has very good resistance and other things. So we are looking after the, okay, this may be the one which we can explore in further, like bringing down the useful alleles and genes to the cultivated ones. So that is the plan. Most recently, I don't have information, but would like to say here, we have here around some 77 accessions of egg glabrata, and we found the very good resistance for late leaf spot and stem rot that we could not see much in cultivated really. And really that is a good source that we are trying now through embryo rescue and other things, and then to get transfer into cultivated one. So that is the long-term plan with us in the pre-breeding program. Okay, if you just summarize this, like say where we have been in 2013, where 2022. So we can say without genomes, we are having very small assays, but now you have everything, like say reference genomes, high density assays. We are doing WGS, performing QTL-seq. We have low density panel. We have here, like say genome assemblies, gene expression atlas, as I mentioned, then quality slips. So maybe you can see some of the applications can say these are for breeding purpose. And I will be just going through one by one what I mean by saying that. So high throughput genotyping assays. For breeding, you need all these things. So we developed in 2017 one high density assay that has 58,000 SNPs. So that is being used mostly for trait mapping, diversity, and all those things. Initially, we used for genomic selection also. But you know, this is not so much cost effective if you want to deploy in the large scale in the breeding program. So we thought that for genomic selection purpose or for getting the background selection done or genetic purity, we should have a cheaper option. So if I give an like say an idea, like if this one we are investing around some $55 per sample or so, here we are investing just $6 to $7 per sample for 5,000. So that is the like we are saying the effectiveness or cost effectiveness for large scale screening. So that's very good if you are getting that much like $6-7, you are getting 5,000 SNP data. That's really like very cost effective. Then we have diagnostic markers where we have agreement with Intertech, and we have kept here we get for 10 SNPs in $3, 2.5 to $3 per sample. So that is like say we use for hybridity confirmation, genetic purity, and also we use for foreground selection. So that is how we are looking for. So what you have seen from the reference genomes, then resequencing, we got SNPs, we made use of those genome assemblies for trait mapping on one side. On the other side, we use those SNPs for developing these assays, and we can develop and customize those assays in future as well. One thing here important to mention here, in mid density, we have kept the diagnostic markers also. So you can perform foreground selection and background selection also. So that's the beauty of this assay. This is very dynamic in nature, and we can replace the SNPs and make it the better always. So that is another opportunity in this one. Earlier, like say some 3 years back, what we were doing, we were planting all the F1s or BC1 F1 plants in the field. And then from this, we were doing the genotyping, like DNA isolation and going for genotyping. But now we are not working in that fashion. What we are doing, if we have BC1 F1 seeds, we chip them, we send directly for genotyping. And then if the positive plants only, that will go for sowing. So suppose if we have say 10,000 F1 seeds, we will chip them, we will genotype them for the foreground markers. And if you found only 3,000, 2,000 seeds useful with those favorable alleles or like say SNPs, then only we will go for planting. So that's how we are saving these resources. So it's a very cheap and seed size is bigger. So here we have advantage. We can use this technology. Now, what are required actually for targeting, like say, I mentioned about feature ready groundnut. So basically we have to keep on adding the traits which is required that we see in future, that is not in terms of climate, but also what farmers are needing, what consumers and industries requiring. So these are the, like you can say, disease, whether it's a leaf rust or leaf spot, heat tolerance, all these things you can see. These are the traits. For biotic resistance, you see then quality here, oil with the protein content and all those things, fatty acid. These are the like quality traits you need. Fish seed dormancy, this is the trait where you can see if you get rain just before harvest, most of the crops will be like germinating in situ germination. So basically in field itself, it will jump in it. So this is really a very tricky trait, and we need to have dormancy for at least 15 days in all the groundnut varieties. So that is very much required. Blanchability, this is like one of the like trait that is required from the industry that we are working on. Aflatoxin contamination, we will not be discussing here much, but I will update on the progress because this is one of the like most complex traits that we have come across in case of groundnut. Now, I was mentioning about that we have in groundnut at ICRISAT around some 15,000 diverse accessions. From this one, we have made a smaller set, that ICRISAT reference set having diversity from this 300. that we sequenced and phenotyped for many traits. You can see here, multi-location trials and for more than 50 traits. But those mostly these are agronomic traits, morphological traits, but also there are useful traits that is required in the breeding program. Now let's go for one of the like one by trait, and then how we are developing those or develop the genomic tools from that. Now come to first high oleic acid. You can see that high oleic trait has become in India actually. It was not so discussed about, but high oleic groundnuts are grown in US, Australia, Argentina on like saying very high percentage. So that is demand. And then high oleic ones, if you are aware, this is very good actually for good. It gives good cholesterol. What we did earlier when I was in University of Georgia, so I was like basically doing their genetic mapping and then estimating the effects of FAD2A and B alleles, and then developing. So and when I continued here and then developed those CASP assays, which are now on Intertech, that can easily differentiate the high oleic and low oleic genotypes. So that is like basically from genes to markers. These markers are available at Internet platform. Anybody in the world can use it. So this is one trait. We have markers and deployed in breeding. The second trait is for leaf rust and late leaf spot together, because these both comes most of the times together on leaf. So we call it together as a foliar fungal disease. In that way, we got like from genetic mapping in A02 and A03, we got two regions. So A02 controls for late leaf spot 50%, and the rest 50% of the late leaf spot comes from A03 from the same region, which controls 80% rust. What you can say, then that means this A03 region of A03 region is most important, say controlling 80% rust and 40% late leaf spot. So that's most important. So we have developed markers from these two regions, and that we identified through genetic mapping and sequencing method, whole genome sequencing, that we call QTL-seq here. And we developed markers also from that. So this is about these two traits. Then again, fresh seed dormancy, we went for say genetic mapping, and here also, and also for QTL-seq approach, so to get those genes and markers. Those are being now available, whether it's lab-based markers. Now we have developed and validated, and it is in Intertech platform. So you can say this is low throughput. You can do in lab if you have less samples, or you can use the Intertech facility where you can go high throughput. You can genotype thousands of samples in a day. So this is about fresh seed dormancy. Then let's go to some more complex trait. And that is like stem rot resistance. It's not so easy trait actually. And phenotyping is very tricky. And it's very like say non-consistent phenotyping data is one of the problem, and which has also reflected in our studies that where main effect QTS means loci directly impacting the trait, we got very of less impactful, but the interaction between the loci, QTL interaction that shows major role. And there was some common reason here we got, which was in for main effect as well as say interaction, a good reason. We got some of the candidate genes and then markers separating these genotypes, but we have not validated these further to use in the further breeding program. We are now what we are doing, we are exploring further for this trait. Let me complete this stem rot. So basically now we are doing phenotyping in reference set that for 2-3 seasons, and then we have WGRS data. So we'll be going for WGRS as well. Maybe we can get better genomic regions and results, and then we will go and developing markers for the breeding program for stem rot. Now seed weight, as you know, even that you can't be able to know even seed size also of groundnut. For other crops you can see because it is seen above the plant, but you know that groundnut seed that is inside the soil. So basically everything is like say magic until unless you take out the seed, you don't know actually what is the size of the seed. So sorry, breeders they told us that, okay, even for seed weight, we need markers. So we went ahead and for genetic mapping and QTL-seq. So KTL-seq, again here, you are hearing again and again, here we are pulling the, like say, extreme set of say 2020 samples from the real population. So that helps us in getting the candidate genes. So here also we developed these markers, and these are available in the public domain basically. And then this paper is recently 1 month before published in the plant genome recently. Now, this is another one, iron deficiency chlorosis. So if it is like there are much of the soils where we have iron deficient soils, and you can see clearly how it shows the symptoms. So you can see phenotyping is very easy for it, because the greener it is like say more of tolerance, while you see the yellowish, this one, this is more of like a sensitive kind of thing. And we got good QTLs here, but yet we have to develop those markers and validate that process is going on. So we are on to this trait. Then again, coming to heat stress, and before this, I should mention about drought tolerance. So because still in groundnut, you don't have a, like say particular say parameter physiological which you can target, like as in case of chickpea where you can target long roots. So that will give drought tolerance. In groundnut, we don't have any such trait which you can target for drought tolerance. So that's really difficult. Getting markers for drought tolerance in groundnut. However, it was good that we had one project from ICR NSF, US Dharwad and RARS Tirupati from Mangru. They are partners in this. Really, you can see the phenotyping happened very good at Tirupati, where the temperature goes very high, and then you can see really the behavior of susceptible and the tolerant parent there, so and the population. So that's usually you can see it's a very nice analysis. We have done analysis for all these parameters related to heat tolerance, and we got, it's a really big, this one we are finalizing now manuscript. But to summary say that for many of the traits under heat tolerance, we could get major QTLs here. And many of the traits we got common QTLs in A03, and then 12th chromosome and also 20s. Now these are the our target now. Chromosome 3, 12, and the 20 chromosome are are now target for us. We want to go for candidate gene discoveries and develop markers now. So, and because we are getting here for many traits up to say 38% in this chromosome, 44 here, and 49% here. So this is now the recent one. So maybe next year or this year, you can say 2023, we will be getting to the QTLs. Maybe something, some advancements may happen for the heat tolerance. Again, for drought tolerance, what we are trying exactly not the physiological parameter, but mostly it's like say yield parameters under drought stress. So we have one magic population which is being phenotyped. You can see for many seasons in Tirupati and Dharwad, and also at Ikrisat, that we are doing QTLalysis now. Maybe we will have results if you want to see. There are some results here for drought tolerance, and some are good QTLs, we can say, where we found, and then we would like to work in this, like say some of the regions to see if something is useful and something can be applied to the breeding program. So you can say still we have to move from genes to markers here for this trait. Again, aflatoxin, as I mentioned here, so you can see that we have done like resequencing for this 300. So 184 is part of that, which was phenotype for 6 regions. We got good here regions here, MTAs, and then we developed CASP assays also. And right now we are practicing actually haplotype based breeding for this aflatoxin contamination. So this program is going on. So maybe we will come to know how much these markers genes are effective in maybe 1 or 2 years when we have these stable pyramided lines. And here for the first time we are using haplotype based breeding. So that's the different from the another like say simple marker assisted breeding. Okay, to take more risk, you might have heard about peanut allergens, and so we tried to see that okay, what is there in store in our diverse germplasm. So we developed earlier protocol was not available from, you can do from groundnut kernel. So what we did, we first developed the ELISA based protocol where we have used kernel. So we can do large germplasm. Then we did 300 core collects our reference genotypes for 2 seasons. And really we identified some of the lines with very low allergen content. So RH1, you can say, and 2 and 3, these are like very, like say, major allergens. And you can see for this genotype, you have only 4 microgram per gram. That is like say below threshold, a person can get allergenicity. There is another genotype, you can see you have 41. So I'm saying for ARH1, this is for ARH2. I mean to say that we have a screen for 300. We may go for larger germplasm, who knows that we may get even like say, even lesser or maybe equivalent genotypes more and more. Okay, what is the importance? We have also phenotyped many of the current groundnut genotypes that we eat today that are cultivated. And you can see how much we are eating, say 15,000 microgram for ARH1. That's huge. Girnar 5, huge. I mean to say here that the current varieties we are eating, they are more allergen. So if we are becoming tolerant, for Indian population may not be that much worry, but please remember that we are exporting now. Once you spot this peanut allergen is very serious for UK, other European countries, and also Australia, even US. And then maybe for trade, it will haunt us. So definitely, I think more work is required in this area. Although we have done exploratory work, but maybe more work required in this area. Now, based on this, what I explained before about different important traits, so far, we developed diagnostic markers for high olic, resistant to late leaf rust, late leaf spot, rosette. That is not in India, but it is more applicable to Africa, seed weight, fresh seed dormancy, and selling percentage. Now these are available, and then we can select 10 SNPs basically. So we can have high olic 2 SNPs and maybe some 8 SNPs from these 2 traits and 10 SNPs. Basically, we can get data for 4 gram selection for 3 traits in just $2.5 to $3 per sample kind of thing. So that is how it is going to work. Maybe in future we will have the efficiency of efficiency information for all these markers. And maybe like rice, if we can get clearly gene-based markers, definitely we can club 5, 6, 7 traits even within 10 SNPs panel, and then that it will be very cost effective. Then in $3 you can select 5 to 6 traits in the segregating populations where you cannot even phenotype. So that will be really a great advantage. Now, these diagnostic markers only not we are using at ICRISAT. You can see who are all using. So you see, these are being used by different institutes from different parts of the world. So we feel very happy that whatever technology we have developed, that is being used from different breeding programs across the world. So we are very happy about it. And then we are collaborating from different institutes across the world for the traits which is specific to them even. We are ready to work and then develop these markers and provide them for use in the breeding program. Okay, what we have done, we have used markers for high oleic acid, and we have improved at least by now around 10 varieties converted into high oleic. From out of them, till now, 2 Virginia bunch varieties, Girnar 4 and 5, are been already released in 2020 in collaboration with ICR, DGR, Directorate of Groundnut Research. And then more this year recently, and these 2 are Virginia bunch. And this year, last year, I mean to say 2022, GG40, that is been released from Junagadh Agriculture University. So together with ICRISAT. So basically now this is Spanish bunch variety. As I mentioned, that Spanish bunch is having the largest area. And now high oleic variety that is being now available, and maybe this will occupy a major area in groundnut in India. So, and it does not have any yield penalty, I would like to state. So if you see, like you have here our like normal genotypes here, where you have around more than 60%, you can see oleic acid. This is the donor that we have used from US. So the trait was not available in Indian germplasm. We use this one to put in different backgrounds, and you can see many of the lines, Girnar 4, 5, Now they have like this kind of situation where they have 80% of higher oleic acid in those lines. So that's a really good one. So now no need to use sun oleic because that is of different botanical varieties that we grow. You can use directly these varieties as a now donor also for developing future variety. And this like initiative even was funded by Mars Wrigley, and then we had from Indian government also funding. And then now we are exploring basically extension for these varieties, and more likely we will have in future this high oleic varieties clubbed with other traits. How? To give an example, we have also markers for rust and late leaf spot, and once you introgress those QTLs, you can see directly impact. This is like say the recipient, a susceptible parent. You see this is a resistant parent. Once you transfer QTL from here to here, the recipient parent becomes like this. So this is a really good advantage of it. Using the markers for these two diseases, we have worked for US Harvard, and Dr. Ramesh Bhatt from their US Harvard has released two varieties having these QTLs in two varieties, TMV2 and then one more variety that has been now released for the state in Karnataka. So that's a good news. Now what we are doing is pyramiding, putting the resistance to rust and late leaf spot and high oleic, and many of the varieties you can see 5-6 varieties are listed here, and then the lines are developed. Now they will go for multi-location testing. So maybe in coming years you can see that varieties with pyramided lines for both the traits will come there, and maybe in future we want to club now with fresh seed dormancy as well. Coming back to aflatoxin, as I mentioned, we are going for haplotype based breeding. We have developed, selected different varieties, mostly here large seeded varieties, because most of the times the aflatoxin is a problem where we want to export. So we have chosen those varieties to be improved, so that if something come out resistant variety or even tolerant also, these can be, the production can be used for either export or export driven products in India itself. Now, after aflatoxin, what you can see, we are also like I just mentioned about marker state by cross breeding, gene pyramiding, and then haplotype based breeding. Now also what we are moving towards genomic selection. What you can see in the slide, we are working with the University of Edinburgh, the Roseding Institute. John Hickey, you might have heard the name from John Hickey. And then also now he shifted from there. So we have now Grieger there, Gorznak, who is taking care. Here, two-part strategy and then multi-part strategy. What is this two-part? Here what we are doing, improving the parents and then developing the products. So here the lines used are elite ones. The another approach is multi-part, that is not being done in any of the crops, but this multi-part genomic selection is used in animals, cattle, like say sheep in UK. So we are trying for the first time how we can deploy in the groundnut. And the process here is that we want to bring the more diverse germplasm into the elite pool for the crossing, but it predicts that how many cycle it will take to bring in. So we can fast pace this process. And if suppose if it is taking say 10 years to breed elite from this one, it may take maybe 4 years to do that. So that's how it is going to work, but we have to see how it is going to give results in coming days. Now, if you see that how we want to exploit genomic selection, it's not just that you just select the plants. You can have multi-part GS strategy for integration of exotic genetic diversity. You can have multi-trait GS keeping in mind of genome like genetic and environment interaction, genotype and environment interaction, and then also you can have say population improvement where you can develop product development. Also, you can develop the or select the parents also. I mean to say that the parents for the breeding program also needs to be recycled in a like say timely manner. So, and then you can have optimal cross selection that helps us in predicting and the better parents. So that we have to do that, and it estimates what crosses you can make. So that's a good approach. So, and the mid density panel I explained that was the purpose for genomic selection. As part of pre-breeding program, we have like synthetics. So these are basically developed using from different genomes from diploids. You can see here using the colchicine, like say treatment, you double, and then this one we are using in the breeding program to introduce the diversity. And what we can see, actually, these are the some introduction lines developed from these synthetics are showing better resistance than the available in cultivated, and these lines are now being used as a parent in ICRISAT and even Harvard breeding program. So this is the another one, and in future we are going to use A. glabrata, as I mentioned earlier, in a big way in our pre-breeding program. Okay, now to summarize, like how we are going to use the genomics in the breeding program. So you have this crossing program, very simple. You have two parents, you cross it, you get F1, F2, F3, and then F6, F7, then you get different trials and then go for release. What we are proposing here, the parents, you need to check the genetic purity you have to do. And then that's why you can use the mid density, and you can always check the genetic purity of the parents you are making the process. At F1 stage, you can use QC panel where we have just 10 to 20 SNPs, most informative. You can use them and then check the hybridity. In F2 generation, you can go for foreground and background selection using the mid density panel or foreground selection markers like this, like say. So it is up to you. And then again, I mentioned these will be like more markers getting, like right now you have for high oleic, rust LLS, fresh seed or seed weight, but in coming years you will be getting more and more markers. So that will, like in rice, you have for maybe some 10, 12 or 15 traits, you can use markers in the breeding program. So maybe this number of markers will increase in coming days. Then again, if you want to deploy genomic selection, yes, of course, it's up to you. You can start with F5, F6, but if you are more confident, you can go to F2 also. But it's up to, for genomic selection, a caution that it's up to you. You have to build up confidence at which stage you want to deploy. Again, we are using the seed chipping, single seed chipping method always, where we can save the resources for genotyping all this. So that is another way, like say, saving the resources. In summary, now in groundnut, genomic resources and then cost-effective genotyping assays, I think these are models, everything, no more issues. So basically you have all these resources, platform set for deployment of genomics in the breeding program. You can use some of the, like say, comparative genomics also opportunities to learn from cross species. And I think we have enough resources to learn and do something in groundnut as well. Then again, the question remains for high throughput phenotyping. So genomics cannot do anything if you don't have perfect phenotyping. So that's basically, again, we are coming to square one. We are facing the challenges for many of the traits for which we don't have any, like say, set protocol, or we can get consistent phenotyping data. So that's an issue that we have to work on. Again, integration is very important. So NGS technologies are there, and then for breeding faster varieties, yes, you can reduce the cycle and time, but that is on cards and you can do. Only now one more question has come, or the problem I would like to mention, that in more recent years, I see that the funding for research has been reduced, and they are investing more on, say, deployment, development. That means they want more kind of, say, extension activities or so something. So, or maybe the technology dissemination, they are investing more than the technology development. So I think we have to convince the donors to keep on investing in the upstream research, so that there is more will be in the pipeline which they can go for dissemination. And if we will only focus on dissemination, then maybe in few years we will not have anything from the discovery to take it to dissemination. So that's like say question. Also, I would like to mention that ICRISAT got Africa Food Prize for 2021, and I'm very happy that I was part of this project that has made that much significant impact in these African countries. And then that this is a good one I would like to share with you. And thank you. So I will stop my presentation here and will take up questions.
Excellent presentation.
Dr. Goel.
Yes, excellent presentation, Dr. Manish. I don't know how many questions can be asked, because if I start asking, it will be too many. So maybe I give others a chance first. Dr. Ratnaparak, you want to ask something?
Yes, yes. So first of all, many congratulations, and you have covered all the areas with diversity, phenotyping, and everything. Just I have one question regarding, as you mentioned, there's a big gap in productivity in some countries. So what are the key issues where for the low productivity in certain countries, and whether these new technologies will be able to overcome and increase the productivity?
So that's where, thanks for asking this question, Dr. Ratan Parke. I'm very happy about it. And maybe I will share the recent discussions that is happening at the, like say in our international collaboration, that sometimes we get like say something get in flow with the technology that come across, whether it's a genomics or anything, that it will solve all the problems. And sometimes we just say subside or maybe lower down our research activities in other areas. Even for genomic selection, you see that we used to say earlier that, okay, you should have genotypic data, phenotypic data, you train models, and then you predict. But actually, is that only the factor we are getting? There is soil factor, there is nutrition factor, there is microbiome there, soil microbiome there, climate there, already there. Until and unless we are counting into all the factors, we cannot predict the yield actually in the breeding program. So that is one. So in genomic selection or any of the like our selection program, we should integrate actually the other variability for other factors like say microbiome, soil microbiome, data, and then climate, soil nutrition, everything. So that may be useful. So because it will help in say giving like getting more much better prediction. That is one. Another part of your question was, what is the reason that we are not getting good yield in many of the countries or locations? I will give you an example in India itself, that maybe if you go to Gujarat, if you go to say Tamil Nadu, many of the parts, and maybe in Rajasthan, in groundnut itself, you get 3.5 tons per hectare. But now again, you go to some unfavorable conditions in some of the districts of say Telangana and Andhra Pradesh, you may end up getting just below 1 ton. Now that's bring down the average productivity. But if you ask the farmers from those regions, they say still this is like say beneficial as compared to other crops, because it is cultivated in so much resource poor like conditions also. So still it is beneficial for them. So that's how we have to see. Actually, the farmer is not going about the productivity or something. We have to see which one is, what is the best option for the farmers.
Yeah.
So this may be the reason, like as I mentioned here, in different like say countries also, we are not giving proper input, or like say agronomic practices we are not following, so that we are not getting actually the exact potential, the genetic potential in the farmer's field. And that is how, if we compare from the developing to the developed countries, they have large plots, fields, hectares of hectares, and they have large scale operations, let's say mechanization. That's why they get like even the profit, even the like yield potential, everything they are getting like say very good as compared to like say developing countries, including India. If you see where we have very small scale, small holding farmers here. So I see many of the things. So we have to see, we have to learn from them. At least we have to not only just breed good variety, not only the seed with good genetics, but good soil and conditions to express that nice genetics that you have put inside the seed. So that also required. So we have to see that.
Sure. Sure. Thank you very much, Dr. Manish. And thanks once again. Congratulations for excellent presentation. Thank you.
Thank you.
So, Dr. Manish, I have a couple of questions. I have actually a lot of questions, but I will only ask a couple of them. So, first is about allergens. So, what is the role of this gene ARAH for the plant? So, allergen is for us, right? But what does it do for plant?
So most of them are the storage proteins.
So there is, if we remove it, it doesn't affect the plant at all.
Okay, so what we have seen till now, so we have done even like say protein content estimation also for this 300 for 2 seasons. And then we initially somebody asked question whether that low allergen content you are getting only in low protein content lines.
Okay.
So we got into that and we see no, there is no correlation actually between the total protein content and then the allergen content. Then again, what we try to see, we have seen, I have not put that information here. We have gone to amino acid level now.
Okay.
To those particular lines where we see the differences in protein content and allergen content, what is that amino acid? But we have to, I think, need more study where you should have a strategy to manipulate the amino acid level if we can do. But of course, many of the allergen content, they are not just genetic, but because of the processing or the growing conditions, it can increase the content. So like aflatoxin, right? You know, very dynamic in nature. So this protein storage, all those things, these are very dynamic in nature. Like you fry it and other things you do, like you lose and you increase the allergen content also. And then also you may be knowing resveratrol content, that antioxidant. So if it is germinated seeds, you have very high content, all those things. And then once you fry it goes. So many of the things good we lose when we fry, but that is how like the crop is.
Ideally, we should have at least 3-4 lectures out of this lecture. So because there is a lot of information which you have shown in short, like aflatoxin. You said that it's complicated, so I'm not dealing with it, but I will like to hear about it. It's a really complicated trait. And one question only in aflatoxins, because aflatoxin is because of the Aspergillus flavus infection, right? So when we talk about genes, are we talking genes from Aspergillus or are we talking genes from the peanut?
Okay, so let me thank you for this question and let me clarify. So what we see actually, we say pre-harvest and post-harvest infections. If you just divide, pre-harvest means the infection you get inside the field. So and then post-harvest, actually it's like more of a management, like you can process like if you dry the produce properly, you storage them, you process properly, so you are not, you can avoid those like say aflatoxin contamination in post-harvest. So the projects we tried that, okay, can we just very much minimize the infection from the field produce? Then only management will work.
Okay.
Otherwise, if there is already infection, right? Even if store with the infection, it will spread. So that is how we worked on only on this genetic component. Of course, what we have seen in our data set, that even the level of an intensity of infection, even in the same field, it is very much variable. Because the infection and spread and aflatoxin content formation itself is result of the interaction between different microbes, soil microbes there. Within that dynamic, we tried, okay, what is the consistency? So you can say we tried to find consistencies among the inconsistencies there during the phenotyping and then mechanism, and tried to find out, okay, if there is any genomics role there, even if it is 20%, we can capture that and we can provide that kind of seed so that we can reduce the infection in the field, and then management can take care.
So of course, there are many more questions. Just I will ask one more small one. You talked about glabrata, and you are saying that there are problems in a set of seeds in glabrata, but you are trying to include it in your breeding program. And then similarly, you have so many synthetics. So do we have any information what is crossing with them, or you have to use bridge species in between to cross them? Because that's going to complicate the whole process. So any light you like to shed on it?
Yeah, thank you again for this. So what we have seen, that you can see the diversity, different sex and species. So we still feel that basically we are living up with the diversity that is being created some 10,000 years back.
Right.
And maybe we are playing the diversity within that. So, and so some of the synthetics being developed, they are being used. But when we have seen traits that we need now, so that is how we land up in the glabrata. Now the question was, glabrata itself is like a different, like say genome. It has like say different say section itself. There will not be cross compatibility between them. But we are making a strategy. Okay, can we go through embryo rescue? We can bring in the segments there, and then we can see whether we can introduce those genomic region in the cultivated background. Of course, it is very challenging. It will change many of the scenarios, but we would like to try if this can help us in even like say improving those traits that we don't have in cultivated or any other deployed species time being. So
have you tried any crosses yet, like reciprocal? Maybe it works as a male better than female.
So that we have not tried. That's what I'm saying, that A glabrata has come into like say in our limelight now. Even like say even global community also, you see very less uses of A glabrata, because it is of like say annual nature. It does not also, you have to maintain throughout the year, and it will not set seed. So mostly ornamental, you see many. I think you will get those. So many of the countries, they use for this fodder and then ornamental for that. So but this is very like say it has some of the lines very good resistance. So we are trying it still, but we don't have yet results that how to use it basically and how to get results. But that has come in our like say focal areas, how to use this.
Thanks, Dr. Manish. So I can maybe ask others if there is any other question from anyone. Okay, Dr. Manish, I actually have a lot of questions. If I can show you my list here, but I don't know if you can see it or not. But if possible, I will like to meet you. If you are coming to Delhi, let me know. I can arrange a lecture here also. And it was an excellent lecture and excellent listening to you. A lot of information. I just hope I can hear you again on a smaller number of topics, because I will like to know more details of your work. It's very interesting. And thank you very much for giving me a very good evening. I think everybody enjoyed it. And I look forward to meet you in future also. So with this, thank you from the GIL platform, because Genome India International second lecture was also successful. So I hope in future also we have listed, we are making efforts towards some very good speakers. So we will continue with that. And I take pride in saying thank you to Dr. Manish from GII side. And I hope to interact with him in future more.
Thank you, Dr. Goel, and thanks to all of you for having patience and listening to me and sharing the like say journey 10 years of journey. And I wish you all a very happy new year. So now I take this opportunity and wish you each of you a very like happy and most importantly healthy and successful new year. So thank you, and thanks for GII platform for providing this like say opportunity to share our research information. Thank you.
Thank you, everyone.
Thank you, Manish. Thank you, Dr. Shalim.